Hello friends, today we will learn about regulation of viral gene expression. First is bacteriophage lambda. Repression of lambda lytic pathway genes during lysogeny. When temperate bacteriophage when temperate bacteriophage when temperate bacteriophages such as phage enter the lysogenic pathway during which they covalently insert their chromosome into the chromosome of the host, the lytic genes must be kept repressed. Lambda phase can either integrate as prophase into the E. coli chromosome via the lysogenic pathway, or it can enter the lytic pathway. Prophase genes are silent while all lambda genes are expressed in the lytic pathway, leading to synthesis of new phase particles and cell death. Both, both repressor and activator CM proteins are involved in the regulation of lambda phase particles and cell growth. Lambda gene expression. Repressor, that is CL gene product, binds to two pairs OL and OR of operator regions, each of which has three binding sites for the repressor. This repressor binding keeps lambda genes turned off in the prophage. The strongest binding site for repressor in both OL and OR are the sites nearest the first structural gene in each operon. Lambda repressor regulates its own synthesis as a result of different affinities for OR1, OR2, OR3. Repressor binds to OR1, which sorts of crow expression enhances another repressor binding to OR2. When repressor is in excess, it binds to OR3, sorting of its own expression. Lysogeny is terminated when lambda repressor is cleaved by a catalytically active REC-A protein single-stranded DNA complex. This allows CRO gene expression and CM protein binds to OR3 and sort of repressor gene transcription without turning off CM. Repressor cleavage also flows N gene expression from PL. Thus, a lambda gene expression cascade is initiated, leading to prophase excision, new phase, and host cell lysis and death. Regulatory proteins have a helix turn motif. Three-dimensional models of CRC, lambda repressor and CAP show that these polypeptides share an alpha helix beta turn alpha helix motif which is involved in specific protein DNA interactions. These proteins occur as dimers which interact with specific DNA sequences via binding of an helical polypeptide domain with a major group of symmetrically oriented recognition site spanning one turn of a B DNA helix. Now come to lambda repressor. Lambda repressor is comprised of DNA binding domain, dimerization domain, and connector region. The repressor has highest affinity for sites OL1 and OR1, so the first repressor dimers usually bind at these sites. The repressor binds cooperatively to sites OL1 and OL2 and two sites OR1 and OR2. When repressor dimers are bound at OR1, OR2 and OL1 and OL2, RNA polymerase cannot bind to R and the gene encoding functions required for lysis are repressed. Now come to lysis version, lysis versus lysogeny. Spontaneous switches from lysogeny to lysis are rare under normal conditions but may be induced by irradiation with UV light. The initial choice between the lighting and lysogenic pathways depends on the balance between the gamma repressor and the coprotein. If the lambda repressor occupies OL and OR, lysogeny occurs. If CRO occupies OL and OR. When, when a temperate bacteriophage such as phase lambda enters the lysogenic state, the lysogenic genes of the prophase are kept turned off by a repressor operator promoter circuit similar to those of bacterial operants. When bacteriophages infect bacterial cells, lytic growth is governed by pre-programmed sequences of viral gene expression. Now come to bacteriophage SP01. Let's know about bacteriophage SP01. Phase SP01 contains early, middle, and late genes. Early genes are transcribed by the host cell's RNA polymerase. An early gene product binds to the host cell's RNA polymerase and causes it to transcribe the middle genes. A middle gene product binds to the host cell's RNA polymerase and causes it to transcribe the late genes. Pre-programmed temporal sequences of viral gene expression occur in bacteriophage infected cells. The first viral genes expressed in an infected cell are transcribed by unmodified bacterial RNA polymerase. Subsequent sets of expressed viral genes are transcribed either by an RNA polymerase encoded by the phage genome or by bacterial RNA polymerase modified by the addition of viral protein. Thank you.